lapel.
Spray drying encapsulation of elderberry extract and evaluating the release and stability of phenolic compounds in encapsulated powders. Welcome back to our channel. Today, we're diving deep into the fascinating world of microencapsulation, specifically focusing on elderberry extracts and their applications in food, pharmaceuticals, and cosmetics. So, grab your favorite snack, and let's get started. First off, what is microencapsulation? Simply put, it's a process where tiny particles or droplets are surrounded by a coating to form small capsules. This technique is particularly useful for protecting sensitive compounds, enhancing their stability, and controlling their release over time. Our study focused on elderberry extracts, which are packed with beneficial polyphenol known for their antioxidant properties. We used 3 different natural encapsulating agents: modified chitosan, sodium alginate, and gum arabic. Each of these agents plays a crucial role in preserving and delivering the elderberry extract. We employed a solvent extraction method using ethanol and ultrasonic waves to extract the elderberry compounds. This process helps to pull out the valuable nutrients from the elderberries efficiently. After extraction, we mixed the elderberry solution with our chosen encapsulating agents and put it through a spray drying process. This technique transforms the liquid mixture into a fine powder, making it easier to handle and store. Now, let's talk about the spray drying process. We used a mini spray dryer, where the mixture is atomized into small droplets and dried using hot air. This method not only preserves the bioactive compounds but also results in a powder that can be easily incorporated into various products. After creating our microparticles, we evaluated their release profiles. This is where it gets interesting. We tested how quickly the elderberry extract and rutin, the main polyphenol in elderberries, were released from the microparticles. The release was tested in deionized water, simulating conditions similar to those in food and cosmetic products. We found that the encapsulating agent significantly influenced the release rate. For instance, microparticles made with modified chitosan released their contents the fastest, while those made with sodium alginate took longer. This means that depending on the desired application, we can choose the right encapsulating agent to control how quickly the active ingredients are released. We applied several mathematical models to analyze the release kinetics, including the zero-order, first-order, and Course-Meyer-Pepys models. These models help us understand the mechanisms behind the release of compounds from the microparticles. The Weibull model provided the best fit for our study, suggesting a combination of diffusion and swelling mechanisms at play. Stability is another critical factor. We stored our microparticles for 8 months to see how well they held up. The choice of encapsulating agent was key here, as it directly impacted the stability of the bioactive compounds. High encapsulation efficiency was achieved, ranging from 92.3% to 99.8%, indicating that most of the active ingredients were successfully encapsulated. So, what does this all mean? Well, the elderberry microparticles we developed have tremendous potential for use in various industries. They can be incorporated into yogurts, cereals, and even cosmetic products, offering consumers the health benefits of elderberries in a stable and convenient form. In conclusion, microencapsulation is a powerful technique that enhances the delivery and stability of bioactive compounds like those found in elderberries. By selecting the right encapsulating agent, we can tailor the release profiles to meet specific food, pharmaceutical, and cosmetic needs. Next, let's discuss technology selection. The article we're exploring highlights several compelling reasons for choosing spray drying technology to optimize drug formulation, particularly when it comes to encapsulating elderberry extract and its bioactive compounds. First off, spray drying is a rapid process that transforms liquid solutions into dry powders in the blink of an eye. The drying step lasts only milliseconds to seconds, which is fantastic for heat-sensitive compounds. This quick process minimizes thermal degradation, ensuring that the beneficial properties of the elderberry extract remain intact. But that's not all. Spray drying achieves high encapsulation efficiencies, with rates ranging from an impressive 92.3% to 99.8%. This means a substantial portion of the active compounds are encapsulated, preserving their bioactivity and effectiveness. Another key benefit of spray drying is that it creates a physical barrier around the active ingredients. This barrier protects them from environmental factors like oxygen, light, and moisture, significantly enhancing their stability and shelf life. What's more, spray drying is scalable and flexible. It's suitable for large-scale commercial production, making it an ideal choice for both food and pharmaceutical applications. Plus, it offers versatility in selecting encapsulating agents. Whether it's modified chitosan, sodium alginate, or gum arabic, you can choose based on the desired release profile of the active compounds. Speaking of release profiles, spray drying allows for controlled release of these compounds. The release kinetics can vary depending on the encapsulating agent chosen, which means you can tailor the delivery of the active ingredients to suit specific needs. And let's not forget about processing losses. Spray drying ensures minimal losses during processing, which is crucial for preserving the efficacy of the final product. In summary, spray drying technology stands out as an optimal choice for drug formulation due to its efficiency, high encapsulation rates, stability enhancement, scalability, versatility, controlled release capabilities, and minimal processing losses. One of the standout features of spray drying is its speed. This innovative process can dry materials in just milliseconds to seconds. This rapid drying minimizes degradation, which is particularly beneficial for heat-sensitive compounds. So, if you're worried about losing the potency of your ingredients, spray drying has you covered. But that's not all. Spray drying boasts an impressive encapsulation efficiency, with rates ranging from 92.3% to a staggering 99.8%. This means that the active compounds you want to retain are effectively encapsulated within microparticles, ensuring they remain intact and effective. Now, let's talk scalability. Spray drying is not only efficient but also highly scalable. This makes it perfect for large-scale industrial production while maintaining consistent quality across batches. Imagine being able to produce high-quality powdered ingredients in bulk without compromising on quality. Another significant advantage is storage efficiency. The resulting dried powders occupy much less space compared to their liquid counterparts. This not only improves handling but also makes storage more efficient, saving you valuable space in your facilities. Stability is another key benefit. The encapsulation process protects sensitive compounds from environmental factors such as oxygen, light, and moisture, which can degrade their quality. This enhanced stability translates to a longer shelf life for your products. Flexibility is yet another perk of spray drying. This technique accommodates a variety of encapsulating agents, allowing for customization of microparticles tailored to the specific needs of the active ingredients. Whether you're working with natural gums, proteins, or other materials, spray drying can adapt to your requirements. And let's not forget about controlled release profiles. Spray drying allows for the tailoring of release mechanisms using kinetic models. This means you can design your products to release their active ingredients at specific times or rates, making them even more effective in their applications. Finally, one of the most reassuring aspects of spray drying is the minimal loss of active compounds during the process. This ensures that your final product is packed with high concentrations of bioactive substances, maximizing its effectiveness. In summary, the advantages of spray drying technology are numerous: rapid processing, high encapsulation efficiency, scalability, reduced storage volume, improved stability, versatility, controlled release capabilities, and minimal losses. These benefits make it an ideal method for microencapsulating sensitive bioactive compounds in both food and pharmaceutical applications. Next, let's us dive into the fascinating world of excipients and their crucial role in formulating elderberry microparticles. When it comes to creating effective microparticles, the selection of excipients is paramount. These are the substances that help enhance the stability and bioavailability of encapsulated compounds. Our study focused on 3 natural encapsulating agents: modified chitosan, sodium alginate, and gum arabic. Let's start with modified chitosan. This remarkable excipient was chosen for its biocompatibility, biodegradability, and low toxicity. One of its standout features is its water insolubility, which is particularly beneficial for controlled release applications. By introducing hydrophilic functional groups, we enhanced its solubility, allowing for a faster release of encapsulated compounds. This makes modified chitosan an excellent choice for applications that require immediate bioavailability. Next up is sodium alginate. This natural polysaccharide is renowned for its biodegradability and low toxicity. Its ability to form hydrogels through ionic gelation sets sodium alginate apart. This property allows for a controlled and slower release of compounds, making it ideal for ensuring prolonged bioavailability. And then we have gum arabic. Known for its emulsifying and stabilizing properties, gum arabic was included for its ability to stabilize emulsions and enhance texture in food products. With a long history of safe use in the food industry, it's also compatible with various other ingredients, making it a versatile choice. In summary, these excipients were selected for their unique benefits, ensuring the stability, safety, and effective release of elderberry's phenolic compounds. Whether in food, pharmaceutical, or cosmetic applications, these natural agents play a vital role in maximizing the potential of elderberry extracts. Welcome back to our channel. Today, we're diving into the fascinating world of elderberry microparticles and their experimental process, parameters, and product characterization. First up, let's talk about how these microparticles were characterized to assess their quality and effectiveness. The total phenolic content, or TPC, was determined using the Folin-Siokalta method. For this, we mixed 100 microliters of the phenolic extract with 2 milliliters of water, 1 milliliter of a 15% sodium carbonate solution, and 200 microliters of Folin-Siokalta reagent. This mixture was then incubated for 2 hours at room temperature. After that, we measured the absorbance at 765 nanometers using UV vis spectrometry, with calibration curves prepared using gallic acid as a standard. The results were impressive. The rutin microparticles showed the highest phenolic content, ranging from 744 to 784 milligrams of gallic acid equivalents per liter, while the elderberry extract particles had lower TPC values. Next, we assessed the particle size distribution using laser granulometry with a Coulter LS230 particle size analyzer. Ethanol, at 99%, was used as a dispersant to prevent any particle agglomeration. Now, let's take a closer look at the surface morphology of the microparticles. This was analyzed using scanning electron microscopy, or SEM. Samples were fixed on brass stubs, coated with gold, and examined at various magnifications, ranging from 1,000 to 50,000 times. Moving on to the release profiles, these were assessed in deionized water for both the encapsulated elderberry extract and rutin. We applied various mathematical models to understand the release mechanisms and kinetics, including zero-order, first-order, Higuchi, Korsmeyer-Pepys, and Weibel. The findings revealed that modified chitosan produced the fastest release, while sodium alginate led to a slower release. Interestingly, the release kinetics were best described by the Weibel model. Encapsulation efficiency was remarkably high across all formulations, ranging from 92.3% to 99.8%. This indicates effective entrapment of the active compounds. After 8 months of storage, stability studies showed that the release profiles remained similar to those of fresh microparticles. However, the TPC and concentration of active compounds released increased, likely due to oxidation and degradation of the encapsulating agents. The yield of the microparticles ranged from 25% to 41%, with lower yields observed for elderberry extract particles attributed to increased adhesion to the spray dryer walls. In summary, these characterization techniques confirmed that the encapsulation process was effective, providing high encapsulation efficiency, favorable release profiles, and good stability. This makes the microparticles a viable delivery system for bioactive compounds in food, pharmaceutical, and cosmetic applications. Now, let's discuss the optimization of process parameters and their impact on the final formulation. Optimizing these parameters in the production of elderberry microparticles through spray drying is crucial for achieving desirable characteristics in the final product. The study focused on key parameters such as the selection and concentration of encapsulating agents, spray drying conditions, and storage methods. These factors significantly impacted the microparticles' encapsulation efficiency, release profiles, and stability. The encapsulating agents chosen, modified chitosan, sodium alginate, and gum arabic, were optimized based on their release characteristics. Modified chitosan promoted a faster release, while sodium alginate provided a slower release. A concentration of 1%, with V, of these agents was used to ensure adequate viscosity and high encapsulation efficiency. Spray drying conditions were also carefully adjusted, with an inlet temperature of 115 degrees Celsius, a flow rate of 4 milliliters per minute, and optimal air pressure and aspiration rates. These adjustments were made to preserve heat-sensitive compounds while ensuring effective drying. After production, the microparticles were stored at 4 degrees Celsius, maintaining their stability over 8 months, with only minor degradation noted. The optimized process resulted in high encapsulation efficiencies, allowing for tailored release profiles that could be adjusted for immediate or sustained release based on the encapsulating agent used. The microparticles also maintained their stability and physical characteristics, such as a regular spherical shape, highlighting the success of the optimization. Overall, carefully controlling these parameters demonstrated the importance of process optimization in developing effective microparticles with potential applications in the food, pharmaceutical, and cosmetic industries. Thanks for watching. If you found this video informative, don't forget to like, subscribe, and hit that notification bell for more exciting content. See you next time.
PharmaPal.